Good morning, good afternoon. So my name is David Rouquier. I'm working for Bayer. I'm based in Sophia Antipolis in south of France. And I'm the pleasure to start to present the status for this OASIS project, which aimed at exploring the cell painting and omics for liver toxicity assessment. On the next slide. So I just started to before Anne joining afterwards to present you some overview about the study and the big questions about this project. On the next slide. So as you will see, this project and the next click maybe will be composed on, it's a multi-sector project. And from the industry perspective, we have both the agrochemical sector and the pharmaceutical sector, which are involved. And with, I would say, two different kind of driving forces and interest into this project. For the agrochemical sector, we are facing the situation where there is a strong call to, if not eliminate, at least reduce dramatically the use of lab animal testing quite shortly. So there are some deadlines going on up to 2035, both from the US EPA, or the North American authorities, but also from the European regulatory authorities. For the pharmaceutical industry, the driving force is a little bit different. So, of course, we are talking about reduction of animal testing. There are some discussion about the FDA Modernization Act 2.0, but the most of the driving force for the pharma is about the gaining information and knowledge about the translation between preclinical and clinical. So the overall vision of the project, as I said, is to have a multi-sector. You will see multidisciplinary, collaborative, pre-competitive effort, where both the specific needs from the agrochemical and pharma sector could generate useful insights in the context of the new approach methodologies. On the next slide, please. Connie?
I moved. One more? Maybe
no,
there's a little delay.
There is a lag. There is a lag in my because I'm still on the slide 32. Sorry, but I try to make it. I will do it. Yeah, let's be flexible. So the mission, but fortunately I have two screens, so I can have the slides on the other on my other screen. So the mission for this project is about gaining confidence on the combination of cell painting, now it's coming, and transcriptomic and proteomic for safety assessment of small molecules using hepatotoxicity as a use case. And for this, the intention is to benchmark already existing in vivo hepatotoxicity data that has been already generated either in rat, in rodent, or in clinical trials. By a series of, in a series of experiments, and to benchmark this toxicity, hepatotoxicity profile against molecular and phenotypic cell-based assay results. On the next slide. So for this, the idea is to make use of public and private compounds with the associated in vivo data or human data. So we are talking about a rather decent high number, still not very high throughput, but still for tox a decent and high number of compounds, around about 2,000 molecules, both from the public domain or donated by the industrial partners. And we are having in mind to test several different cellular models, increasing the complexity of the physiological relevance of those assays. So starting with cell lines, moving to primary rat hepatocytes, organoid, and liver on the chip. And in terms of technology, we have the great privilege to have with us Anne Carpenter and Shantanu Singh, who are the true inventor of the technology. We will be also doing some mRNA profiling, so transcriptomic analysis and protein profiling. And out of those evaluation from all those technologies in the different cellular models, we will generate a comprehensive data analysis pipeline and workflow to be able not only to determine some dose response evaluations, but also translation of in vitro to in vivo concentration, the so-called key-viv aspects, in order to make the move between in vitro to in vivo. And as I said from the beginning, this is a pre-competitive project, since the intention is to really go into to make everything generated into the project fully accessible to the public, and also the methodologies. On the next slide.
Should I click through?
Just yeah, let's go through click up to the yeah. So we're gonna, so this is the maybe this slide you have seen that many times. So it's adapted from the NRSC Council, so the toxicity testing in the 21st century, published back in 2007. So when we will be talking about different kind of chemicals. So as I said, both agrochemicals and pharma. For the in vivo biological species, we will be talking about human and rat liver effects, adverse outcome, both from the agrochemical and the pharma sectors, because this type of biological species can be seen as being different. And all the intention is to investigate these in vitro biological species, as I said, using cell painting, tempo-seq for the transcriptomics, and ELISA for the proteomics, all the time in those response to, with the next click, make all the knowledge what one can do, another one on those different cell line, and make all the learnings between these two species, the in vitro biological species with the in vivo biological species, both in human and or in liver. On the next slide. So the anticipated outcome. So obviously a novel data set on rat and human cell that inform cell painting practices for toxicology, molecular data for hepatotoxicity screening and or safety assessment of chemicals, a new multi-platform informatic methods for predictive toxicology. So we are really fully into this system toxicology topic, with an expert network of toxicologists, cell biologists, omics experts, and bioinformatics, so across different sectors. So really multidisciplinary project, which will generate, of course, publication data and databases and platform for pool resources. On the next slide. So who we are. So here is the leadership team. I'm very honored to be the co-program chair together with Dr. Anne Carpenter. And then you see there are different representatives from the public sector, from the agrochemical sector, from the pharma sector, with the great support for the team from HESI. And on the next slide, the status for the current partners at the moment. We are still recruiting. We try to still expand the number of partners into the consortiums. So ranging from academia, government, non-profit, tech partners, which are going to support us in some technologies, in particular proteomics and organochip, and also the industry partners, which are listed there on the right-hand side of the slide. And on the next slide.
All right, I'll take it over from here.
Yeah, I think it's now your turn. Yeah, Anne, please go ahead.
Hi, everyone. I'm Ann Carpenter from the Broad Institute, and I'm helping to lead the project. One of our biggest contributions was writing a grant last fall, which we're happy was awarded, not publicly announced yet, but it is more than $1 million that is earmarked for creating large data sets. And it has to be done in Massachusetts. So we are based at the Broad Institute. Shantanu Singh is here. He's the principal investigator on this grant, and it will allow us to make the data that will be necessary for the OASIS project, at least a good portion of the data. We can advance. One of the requirements of writing that grant was that we had some commitment from a commercial partner who would support data analysis. So the grant pays for the data, and then we need industry partners to pay for data scientists to work on the project. That allowed us to gather groups that could become part of HESI and eSTAR and also contribute to this project directly. You can see it's a $50,000 a year commitment to allow us to put together the data scientists. Next slide. In addition to those financial contributions we've just described, we also have the in-kind contributions. This is really a condensed list of several companies that have expressed their strong willingness and commitment. We have many other partners that are excited to contribute, and now it really comes down to the steering committee and work streams of the project to figure out what would be the most promising different platforms that we may want to test in the project, and how can we leverage all these different potential contributions by different groups. So those are continuing ongoing conversations. We're happy to hear more ideas of what would be useful to add to the project. Next slide. Together, all of the resources that the $50K a year from the industry partners and so on will contribute to the experimental work, which significant part of that is gathering up the compounds for the projects, as well as creating the samples and extracting the data. And then a full-time data scientist at the Broad is going to be split between two postdocs. As many of you may know, one or both of them, Srijit Seal and Jessica Ewald. Srijit's already begun, and Jessica will begin in January and be overseen within our lab here in the Boston area. We also will have a PhD student that's overseen by the other co-chair, Dr. David Ruquier, as well as Professor Van de Waal at the University Côte d'Azur. And it will also need to contribute to the funding will also need to contribute to the storage and publication costs and so on. Next slide. Now, as for the different work streams that have been busy with, I don't know if it's a strength or a weakness that we've had so many meetings so far for this project, but we certainly are making our way. So one of the biggest aspects is selecting compounds. It's not just a matter of dreaming up all the compounds we might like to assess here. We're really quite limited by all the compounds in the world that have some amount of organism level data, rat or human liver tox data in particular formats and so on. So here Srijit Seal's made major efforts to figure out what are all the data sources that exist in the world that are out there in the public, as well as private partners who are willing to share their data for this purpose. And our goal has been to get all of this information about these compounds in a decent, machine readable, nicely structured format. Next slide. So far, we have searched 29 different databases to cross-check and figure out which compounds might be suitable for the project, comparing the overlap across all of these. In the end, you can see the numbers here of what we've accumulated from different groups. Now, in addition to the publicly available data, which is what the main data science was here to figure out what's out there and what's available. In addition to that, we will have access to the eTox private database for a couple hundred compounds. As well, any industry partners are welcome to contribute compounds that have rat in vivo data. So anything, we don't need the chemical structure necessarily. In vivo data that goes along with compounds that can be donated and tested. tested in this compounds profiled via all these fancy methods.
Sorry, was that, and you cut out just for a second? I think it's okay now. Was that for others, or is that just an issue on my end?
No, I heard it too.
Okay, but I think it's good now. It worked itself out. I'm sorry to interrupt you.
Yeah, no problem. And in particular, I want to emphasize this is a project that emphasizes agrochemical and pharmaceutical compounds. And so it's really valuable for folks who want to contribute that are really of interest to you and relevant to your development pipelines. Next slide. Can we go to the next slide?
I'm there. Do others see it? The OASIS study overview? Okay.
Yep. Okay, so in terms of study design, of course, we would love to do every possible cell model that's relevant to liver and every possible high dimensional readout assay type that you could do. So that is extremely impractical in terms both of the finances of sample preparation, but also in terms of the amount of compound that is available for a number of these. So what we are planning to do are for some of the less expensive and less compound consuming methods like cell painting and some of the cell lines, we can do a full scale, all 1,600, 1,800 compounds. But for some of the others, we have to be a bit more judicious. And part of the emphasis of the study design work stream is to make these tough decisions about what subset of compounds shall we put through the more expensive and or more compound consuming methodologies. And you can see that depicted here. Next slide. The study design workstream, as I said, has to make those tough decisions, figuring out which cell types to use, what dose ranges and increments do we want to use, figuring out the assay types and selecting the fancier cell models such as liver organoids and liver on a chip, and basically figuring out which compounds are going to go through which of these different assays and models. Next slide. So far, the study design work stream has gotten cell painting nicely in order. Didn't take much effort to figure out what we wanted to do there, but then assay development to work on the rat primary hepatocytes and the HEPA-RG cells is already in progress, and then eventually we'll get to the methods like organoids and liver on a chip. Transcriptomics, we have a verbal agreement so far with BioSpider to use their platform for the omics on, again, a subset of the samples, and likewise for proteomics. Next slide. Now, what's coming up next are presentations. I'm sure a vast proportion of this whole OASIS project members are going to be at the Society of Toxicology meeting, as well as a couple of other meetings will be represented with different posters just to make sure that the community is aware and that we have access to people who might want to contribute their expertise and knowledge to the project, as well as potential in-kind contributions. Next slide. And we have a couple of papers that are in percolating and pondering how to put them together and drafting. And we, in particular, the process of even identifying what are all the compounds out there in the world that have liver tox available data itself may warrant a publication. That's it. Happy to answer any questions about the OASIS project.
Thanks, Anna and David, for your presentation. We have plenty of time here for anyone who has questions on the consortium. You're welcome to unmute or write into the chat. Yes, Daniel.
Hi. Yeah, I joined just a little bit when the talk already started, but it definitely is interesting. First of all, I'm Daniel Jenner from Maastricht University, and I'm involved in the eSTAR project, carcinogenicity. So I'm involved in bioinformatics and omics data analysis using transcriptomics. So I'm definitely interested to learn more about OASIS and see whether I can participate or not. So who am I to contact? That's basically the question.
Yes, I will be the contact, but I can write your name down now and send you more information after the meeting.
Perfect.
Thank you for your interest.
There's a question in the chat, how many compounds have both rat and human data? I know it's a relatively small set, but I don't know the number. Does anyone else in OASIS remember?
Yeah, I think so. In principle, all the list of the compounds from the DILI list, the DILI rank, sorry, list, or it's round about 1,000 compounds have some preclinical data available somewhere. Have some preclinical data existing. So let's put it like this. Now the key question is to have access to those data, and the first ideas and the first investigation show that we have few hundreds of chemicals for which we will have access to this preclinical data in a 28-day study in the rat for those compounds, which are drugs which were given to a human being.
Leah Weymouth in the chat also asks, any plans to incorporate ecological species to the screens? No, just because of course, financial limitations. We really wanted to stick to those that had a decent amount of public data available, which I believe restricts us largely to rat and human. Our hope is that once we work out with which methodologies reveal which kinds of mechanisms of toxicity, that if the basic strategy and the approach works well with a given assay and a given level of cell model in rats and humans, one would hope that it could also be translated to other species, but that would certainly be a project for follow-up.
I see Peggy has her hand up.
Yeah, can you hear me okay?
Yes.
Okay. Hey, first I want to applaud both HESI and on this activity. I've had interest in this area for some of you that know me for a number of years, and it's been such a big nut to crack. There's so much data out there that we need to be able to pull in to do this, and I think this looks like one of our best chances of being able to pull forces globally to come in and to tackle this. One of the questions I did have, and I was so glad to hear about Anne's discussion on the data analysis, because I think that's going to be a huge aspect. Are we also going to be tapping into artificial intelligence and any of the newer technologies available to us? That's one area. And then I also was curious about some of the other HESI committees that have been looking at, so say data for carcinogenicity for the liver and genotoxicity. Is there an angle of being able to bring that data into? Because I know there's been intensive efforts over the last few years.
Yeah, certainly the project has the expertise of several machine learning experts, Andreas Bender from Cambridge, Oxford, as well as Shantanu Singh, the PI of the grant. His expertise is in that area. That being said, we want to be really careful because the biggest limitation of this project, this field overall, is limited amounts of very precious in vivo data for both rat and human. It's quite limited. And with many machine learning methods, it's quite easy to build a perfect classifier just by overfitting to that data. So we want to be really careful and judicious about how we use our data, and stick to simpler methods where those methods may be effective. I would say that's our first pass plan, is to start with the simpler methods and see how far we get in order to avoid the potential problems of overfitting.
And as far as the other data that you might be tapping into from some other HESI committees, have you guys canvassed the other projects out there amongst HESI committees?
We have not yet done that, but I think also, you know, Carol dropped a comment in the chat, which I think gets to this point, which we could use this as a test for the transcriptomic biomarkers that we're going to hear about today. So I think that's a really good point, and as we go through the meeting, we can talk about that.
Can I just add,
yeah.
Peggy, for things like the carcinogenomics group, we can definitely leverage that we gathered so much of when we already have started to, of the TG gates and drug matrix data for both the list of compounds, you know, are they repeat dose? And that might be something too, that might be an effort of comparing some of the transcriptomic data we generate versus the transcriptomic data that's already out there. So definitely taking learnings from things like the carcinogenomics group, the POD group, how to actually come up with, you know, these the PODs from the different endpoints. So definitely trying as much as we can to speak across the different projects and leverage what was already done. Thanks for raising that.
Perfect. Yeah, I hate to see you reinvent the wheel when there's so much data out there already.
Yeah, and I'll add one more aspect to this. For the proteomic profiling, we will be profiling on the order of 200 to 500 proteins, and the company Nomic Bio that is organizing that and donating that in-kind effort are very open to putting whatever proteins people think are valuable and interesting into their panel. So the next few months, we have a window to propose what proteins get into that panel, which could be informed by the transcriptome and biomarker project or any other information that people have. So we would very much welcome thoughts on what proteins to include.
Thanks,
Great,
Anne.
thanks.
I think there's another question for you, Anne. Will cell painting address only morphological changes, or will it also include protein identification?
I'm not sure what protein identification means in this context. The cell painting assay has 6 dyes that are imaged in 5 channels, and those dyes are dyes. They're not antibodies to specific proteins, but they include concanavalin A, wheat germ agglutinin, a nucleic acid marker, well, HESI, as well as an RNA nucleic acid friendly marker, mitotracker for the mitochondria, and so on. So we won't be measuring specific protein levels using imaging. It'll just be the original channels. That being said, we have all kinds of evidence from all kinds of different projects that we can capture something like 45% of the mechanisms of action classes of approved drugs using this one assay with this set of dyes. So it's you know, not completely comprehensive, but it certainly captures as much of the mechanistic cell states as mRNA profiling does, for example. So it should be good, and we don't expect it to capture everything, but it will capture a fair bit.
Thanks. Chris, you're next.
Yeah, thanks for that presentation. I think I discussed this with David at SOT last year, but you know, the carcinogenomics group is taking advantage of a number of knockout rat models that may be useful for the interpretation of effects that you're seeing in the wild type rat hepatocytes. Maybe in some targeted studies, it would be useful for you to look at responses in the absence of, you know, the major transcription factors which mediate steatosis, carcinogenicity, cytotoxicity, and but they're not available forever. So in other words, where we're taking advantage of in Vigo's colonies, and once we're done, then the colony is put down, and so there may be some timing issues. But I think it may be worthwhile to consider doing some targeted studies with rat primary hepatocytes from those knockout rats. And it may also be useful to do some comparisons in knockout cell models too, for, you know, key transcription factors to help in interpretation of your cell painting data.
Yeah, I think it's like we discussed, it's a very good idea. The thing is that we need to be sure that in terms of the budget constraints, we will have the possibility to think about that. But maybe it's something we could discuss at the leadership team of OASIS, what could be done and what, I don't know what you think, Anne.
Thumbs up.
Yeah, I think something with this project to note is that since we are in the early stage, we're prioritizing where our efforts go right now, but we've definitely started pulling together ideas for maybe a phase 2 or a phase 3 down the road. So I appreciate all the ideas, and we'll keep them under our belt for discussion later. I see Kamen has his hand up.
Yeah, hi. I really like this project. A comment, and maybe it's a question, more of a comment. So extrapolating between the in vitro from a point of departure standpoint, extrapolating that in vitro POD to an in vivo one, I think there are, you know, questions around what's the best way of doing that. And I wonder, you know, if you thought about that, or if you thought about doing some kind of a project, an ancillary project, or working with other groups to really optimize that IVE process.
Yeah, I can just provide a few elements, and you can complete maybe Anne afterwards if you like. So we structure already the project with different working groups, and so we kick out some of them. One of them still not is about the Kiviv topic. And everything related to that, I think it's going to be how to determine the PODs, and how to make use of the PODs to make this translation vitro vivo. It's in the making. We know that it's a key and important aspect, both the determination of the POD and the way to estimate the exposure in vivo. And so it's part of the recruitment we are trying to make for having some key experts in the domain, just to make sure that we do the right things. Yeah.
Yep, I don't have anything to add to that. I can take one of the questions from the chat. What morphological changes are we looking for? So the cell painting assay that we're using, I know this is a mental shift from how many people think of microscopy assays. Normally in a microscopy assay, you design a particular biomarker or protein or something that you're looking for, and you measure that outcome in a very defined way. The concept of using cell painting is actually really much more like transcriptional profiling, where you're more concerned about the pattern of changes that happens, even if any one particular change doesn't necessarily biologically make a lot of sense to you. It's not something you thought you should look at, and yet when we capture images, we extract usually 1,000 or more features from every individual cell, and the pattern of features as they change can reflect changes in the cell state. So it's a mental shift of how these metrics are used. Now, it's true, some of the features are very nicely interpretable, you know, the size of the nucleus or the texture of the mitochondria, how stringy are they versus how blobby are they, these kinds of things. Many of those might be interpretable, especially in the field of tox. And of course, we'll pay attention to those, but I just want to set the understanding that the way we use image data is more as a fingerprint of cell state, and we're looking for, out of all those features, is there any combination of features that's associated with particular types of toxicity, even if we can't interpret exactly the underlying biological meaning of those individual morphology changes. So I hope that's helpful. There's another question, why use HEPA-RGs, cell line in the study, as opposed to using, well, either other human hepatocyte cell lines or just raw human hepatocytes. Now, I think to answer the question of why not just use sort of fresh primary hepatocytes from humans, they are more expensive, but I think it's only maybe 2x more expensive. So I don't think it was entirely the cost. I think it might have been the reproducibility was a concern among the members, but maybe somebody remembers more.
Yeah, this is Heidrun. It's the donor diversity. So it's the reproducibility to some extent. Yeah. And you need to test the different donors. Some played better than others. And if you have tested once for the HEPA-RG, you are fine with them.
To answer another question in the chat, we do, I think the current plan is to use a single time point for all of the assays. Simply, of course, a financial decision. We would rather test a decent dose range and, of course, a decent number of compounds as opposed to testing a lot of time points. We've done some small experiments to compare a kind of shorter versus longer time point of compound exposure, and generally found, you know, of course, there will be some things that you only catch early, some things you will only catch late, but we haven't found it to be a dramatic difference, at least for the time points we've tested for compounds. I believe the plan here is to use a 48-hour time point of exposure for compounds. I believe for, I don't know if that's most of the assays or just cell painting, but that in the past, we've compared that to 24 hours and found not too much of a difference for image data, at least.
Yeah, that's correct. The other question, have we thought of partner with risk hunters? That group has come up, and they are on our short list of contacts to reach out to. Thank you for that suggestion.
Yeah, I think, no, they are already there.
Oh.
Yeah. So yeah, maybe it's the opportunity to highlight another dimension for this project. It's, you have seen maybe that in the partners there are some partners from the regulatory bodies in US, in fact, North America mostly, US EPA, US FDA, but also some representative from regulatory agencies for drugs and agrochemicals in Europe. So that's also part of the ambition of this project, is to make the two sides of the Atlantic talking more together. And Bob van der Waters, the PI of Risk Hunter, has expressed and is part of the list of the people. Is not yet fully active into the thing, but is fully aware about the types of activities we are having into OASIS.